And Julie Heinlein and I are co-hosting today's seminar for Kalina, which is our great honor, of course. Kalina is a professor of biology at Georgia College and also the Georgia Endowed Professor there, or Georgia Power Endowed Professor of Environmental Sciences at Georgia College. Kalina got her PhD at Michigan State University, I'm happy to say, and in zoology and the EEBB program, which is an ecology, environment, ecology, evolution, and behavioral biology interdisciplinary graduate program. She came to the U.S. from Bulgaria, working with Dobrina Tomaskova, right? Topolova. Yeah, and getting her master's degree at Sofia University, and then came and worked on diatoms, and has been the most probably enthusiastically and diversely engaged diatom person that I know, right? So she works in all kinds of different areas, just recently publishing a book on diatom motility, but she's been involved with molecular work very early on, and ecology, and of course taxonomy, even diatoms and medicine, right? You just had a review article a few years ago. So at Georgia College, she teaches a lot of undergrad and graduate students, and teaches the ecology and systematics of algae at Iowa State, Iowa Lakeside Lab. And at home, she enjoys a wonderful husband, 2 kids, a new daughter-in-law. And with that, look forward to hearing your talk, Kalina, on 10 years of diatoms research at the Savannah River.
Well, thank you so much, Jen. I'm honored to be invited by the Diatom Web Academy and to see everybody coming. Just from experience, if you would please switch off your mics so we don't have any background. And you're welcome to stay with your camera on if that's something that you prefer. So it's interesting that, Sarah invited me, and I was just doing some counts for 2021 Savannah River, and I'm like, oh my God, I've been working for 10 years in the Savannah River. I tried to point to you where I am right now. So right, this little blip behind Georgia College is where I am. And this is about 4 counties away from the Savannah River. And it's been important where my location is for some of the research that I'll report to you. And of course, we'll talk about diatoms. And let's see. So there's some diatoms that we'll see from the Savannah River. So just the plan for today, again, why the Savannah River? Why not? I mean, it's absolutely amazing. Even back in Bulgaria, this was so interesting and romantic for me to think about Savannah River. I found this map in the Library of Congress, and it's just so well put together. And actually, one of our location at Port Wentworth was actually very well shown there. There's been a lot of diatom work done, being on the steps of Ruth Patrick and some of the previous researchers was absolutely amazing. And then, I don't know if Dr. Sams is on the phone call, but her and I, 2009, were on something called Georgia Energy Board. And we were told that the Savannah River estuary is going to be deepened and widened, which is now completed. And that's the Savannah Harbor Expansion Project. And I was horrified listening to it about it. And I was like, oh my God, let me start sampling. So 2011 is when we started sampling at location at the harbor, and I'll report some of the data. We've been sampling seasonally for 10 years data there. And of course, I'll talk about a little bit diatom taxonomy. It's the Diatom Web Academy. It's the place to really show off some amazing diatoms. I want to show the power of community analysis in microbial organisms and the messages that they're sending us that hopefully we can understand in many, many ways. So the Savannah River is just amazing system to work with. Upwards right here is a beautiful forested area, and that's where the Savannah research area for 50 years of reference sites it is. And then I'll take you down to the Savannah Harbor and to the estuary. And I also want to show, it's been so much change in our science in the last few years, but something that I've always loved was Thomas Lovejoy, the Patrick principle, looking at ecosystem and just looking at who is there, the species diversity and abundance, something that I've done all my life and my students have done. So I want to start with the Savannah River. So I'm taking you first to upwards in the reserved area, north of the Savannah River, and I'll mention some of the work of former and current students that, of course, have done most of the work that I'm reporting to you today. So the Savannah River site, for those of you that know, is just an amazing place, 1951, that was established by the Department of Energy, and that was a place where streams can be without any alterations from human activities. So this is one of the continuous sites in this nation where we have the entire watershed completely protected within the Savannah River site, but also it's protected, so very hard to get in and sample there. There is the Upper Three Runs stream, one of the biodiversity hotspots of the world. There's been so much research done there, including, of course, the research from Ruth Patrick. And what was absolutely amazing is the persistence. So Rob Mosley wrote a proposal and was allowed to actually go and sample at the Upper Three Runs at the same location where Ruth Patrick actually worked. The goal of his paper was something different, but his paper became quite important for the followed research. We were able to do some chemistry, and we proved that yes, October 2011, the nutrients and diol were sufficient for what we call a reference stream. Rob Mosley took phycology at his senior year, and I was very proud of him that he was able to go to species level identification. He reported to Onosha Carolina, Onosha rhomboidea, unless Paula Fury changed the names. This is what we called them back then, and they were the dominant in the Upper Three Runs Creek. Rob also reported a very large population of Luticola geofertiana. What was interesting in this research, we had two types of samples, so composite vegetation and then more sand sediment sample. The species richness was fairly low. And one of the reasons probably for the very low species richness was, as we'll see in a minute, Gomphoneema parvulum. But after the usual 600 counts and inspired by some of the work that we did with Jen in Kentucky, we continued until we didn't find anything new as morphology that we can all name or taxonomy. So we kind of calculated and saw what was the maximum Shannon diversity for this site can be, and that was significantly higher. In the 600 count, we had 23 or 27 taxa, but we went 150 and 170 when we actually looked at the taxonomy. But why the number were fairly low is probably because of Gomphoneema parvulum. At this point, we were looking at Gomphoneema parvulum sansulato. This is probably today Gomphoneema exilissima. But still, the Diatoms told us the story that we kind of expected. We looked at the Trophic Diatom index, and they were as expected for the area, and the percent of tolerant to organic pollution taxa was fairly low in the Upper Three Runs. So, of course, stepping on the shoulders of the amazing Ruth Patrick, who worked at the Upper Three Runs for many, many years, we were wondering why our data was with so few species richness. And so looking into the literature, this was an image that I really loved as she was designing the diatometer. We actually, with some reports from the Academy, we found out that actually she had diatometers at the Upper Three Runs that she employed. And also I wanted to mention, being 50 years from the Clean Water Act, she was really instrumental reporting to Congress for the Clean Water Act at that time. So we were also able to borrow from the Academy 1956 collection from Upper Three Runs. This was Charlie Reimer collection. And of course, we looked at this exact location. And the data that we were seeing from the Academy was showing us tremendous diversity. So this is the 1956. So at best, we have about 170 different species there. And of course, they were looking at different protocol. They were not stopping at 600, but still the numbers were quite higher than what we found in 2011. And then the amazing Katie Johnson came to my lab 2018. She knew Rob's work, and also she knew that there were diatometers placed in upper 3 runs. So she got the idea, she wrote a proposal, and she was allowed to go place diatometers and then go in 3 weeks and collect them. And actually we had left, right diatometer, and of course we repeated composite samples from similar to what Dr. Patrick and Rob Mosley have done. And it was quite interesting research. Stay tuned. We're working on a paper. I just want to highlight some of the findings that she confirmed. So from the slides that we borrowed from the Academy, so Katie applied new taxonomy, and we found 84 species richness. Luticula reported in 56 mutica was actually Luticula jopertiana that we confirmed. And also 2018, her composite sample was dominated by Eunotia rhomboidea and Gumphonema parvum sensulato. And Katie spent 1.5 years really going to the lowest possible identification within the sensulato Gumphonema parvum community. And this is Luticula jopertiana from her master's thesis. And then most currently, I have Sydney Brown. She's the current master's student in my lab. So we still have the data from the 1956 exact location. She looked at Rob's sample and Katie was able actually to isolate some Gumphonema parvum. We have a living culture in the lab from upper 3 runs. And Sydney looked at biovolume, biosurface, did some amazing population ecology type of study within the Gumphonema parvum from this very important location for us. And we were also able to sequence the 23S and the chloroplast genome. We had 100% what's been reported at GeneBank, and we're working actually on the 23S, and that's a combination with Jim Yeno and Bruce Cahoon from Virginia Tech for some of the collaborations that we have. So it's been very productive work at the upper 3 runs. Now I'm going to take you down to the Savannah River harbor and look at some brackish marine taxa and some very interesting ecological story that we have there. So as I said, 2009 is the first time I heard that the harbor is going to be deepened and widened so we can compete in Georgia, as proud Georgian, with other ports at that time. And listening to what was reported, so there was some money that started to be collected from 1997. They actually started digging and dredging 2015. So it was good that we started to collect 2011. So we were able to have 5 years prior as they were dredging samples, and of course 5 years after, and hopefully we'll continue to collect some data. When I heard about this proposed extension, I wrote a proposal and I got some money. So as you saw, I'm in the middle of Georgia. So for us to go to the Savannah site and collect, it's an expense that I had to plan for all this time, but it was presented to Georgians, they voted, and it's now a reality that we can see what are the effects. And as I was proposing, I was thinking, what are diatoms doing in a this amazing wave of tides coming with marine environment, and then of course the Savannah River bringing all this freshwater taxa. So what we chose to monitor, and again, there's a trade-off. I wish I can go and sample all locations, but again, for practicality, we sampled at one location. It was a USGS Port Wentworth location. And so I'll show you some of the advantages of us looking at that location. And as I said, seasonally we collected since 2011 for all the years. And of course, this location is tidally sensitive, and there's tides that come every day. And also, of course, it's one of the widest places where the Savannah River is coming. So it was a very interesting mixture. We looked at, of course, at high tide and low tide. The state of Georgia is documenting very well, so we knew exactly when to expect. So we timed, we'll leave 5 o'clock so we can be there at 8 and sample at high, and then 12 hours later at low tide on the same day. And we sampled, of course, mud samples. So this is from where we sampled the location. And that's the Fort Wentworth Savannah River sampling sites for us. So the advantage of sampling at a USGS monitoring site, we had any data you can imagine. So this is some dissolved oxygen. They didn't go back to 2011, but we had our own measurements, and they were verified with, again, the USGS measurements, some pH data. What was interesting over the years, precipitation has increased. So 2011, 2016, and now 2021 is the data that I'm going to be reporting. Those are the 3 periods where we kind of concentrated. This is salinity, obviously very important for diatoms. And I'm just going to highlight a few of the works that we have at that point. So the first one that was sampling 2011 was Joseph Domini, and he created this base kind of sampling for us. So high and low tide at the location. We did some experimental work. We looked at top 5 and every 5 centimeters within the high tide in the Savannah River. And what you're seeing is the mud samples that we sampled at low tide. And quite a lot of diversity. We identified very interesting, and I was very surprised seeing all these planktonic, of course, taxa in our mud samples. And I'll mention a little bit Cyanobacteria belgica in a minute, because this was dominant through the seasons for the 2011 sampling and 2016. Slight difference with the 2021 sampling that I'll show you in a minute. We did a lot of collection and work. It allowed me to go back to some of the marine work that I've done back in Bulgaria, and some of the taxonomy, and keep up a little bit with the taxonomy of marine and of course brackish taxa. We did over the years always live and of course cleaned and then SEM analysis. This is Matusiero lawrenceana from the mud samples, very happy and alive, as shown here. There's Campylosauria symboliformis. Again, we did some population studies, some diminution theories, and of course we looked at their physiology. There was some freshwater, so Leucosphaera granulata in the sediment and in the mud was coming from the Savannah River, as we had evidence upstream about it. And again, some very interesting centric, like Cyclotella littoralis, very happy and alive in the mud samples. And we did some, again, measurements and descriptions, kind of complementary descriptions with what's been published in the literature that we have today, some interesting STM work. Some taxa, we were not able to prove that they were physiologically active at the mud samples when we collected them, but they were, again, anything that I'm reporting was at least 5% in any of accounts that we had. Delphinae sororella, very interesting taxonomy. I don't have time much to go into the interesting occlusions that we were able to report within the same sample with what we called sororella, Delphinae sororella, but probably something different. Some interesting taxonomic work that we'll continue to work on that. And then some Pleuroxera levis. I think I've showed it to a few of you. So very interesting ocelli that we saw in the mud samples that we have there. So at that point, 2015, I had Jelena Andresic, similarly enthusiastic about diatoms, come and she went back and recounted everything from 2011 to 2015, anything that had preserved as live samples. She actually looked at chloroplast structure, chloroplast biovolume, surface, and at volume of cells and chloroplast. Did some very interesting work. And what I'm showing you here is 2011 and 2016. This is brackish, freshwater, and marine. And then we separated them based on published literature and what we had into our list on planktonic versus benthic diatoms. And what was interesting in 2016, brackish and freshwater benthic were almost disappearing. That was interesting signal. And then I had another student, Alyssa Thompson, actually continued to do some of the work. And she verified that that's what was happening. They were replaced with planktonic taxa. This is an interesting slide. I don't know if you can see my red arrows here. So there's this tiny thing, mini discus, and then there's the Cimatosira belgica. And again, I love the taxa, and we did 600 counts for all this research, and this was popping time after time, 28% dominance by this little mini discus that we called Chilense, and then of course this Cimatosira belgica that we had in the mud, obviously sieved through the mud from the marine overlay with the tides and the salt environment. So we did some taxonomic work on Cimatosira belgica. It was new for me. It was just amazing to study it. And again, we looked at this very interesting ocelli, so they're kind of elevated a little bit. I don't know if you can see it right here. They're elevated and they make those chains, and probably that's why they were able to survive in the mud. So there's the elevations right here with the you know, those ocelli, and then very interesting. I was seeing it in every sample that we see, and then the students were confirming it in those chains that were just not breaking down that we had there. So we did a little bit of research on that, actually found it quite larger than some of the reported in the literature, and variable in the size in our samples that we had. This one, I mean, I showed you some LMs, but they're so tiny. They're 2 to 3 microns, and we did a lot of taxonomic work, and we went with Minidiscus chilense with some of the SEM work that we did, but this was very, very surprising. So this tiny one, so you need a very good microscope. This tiny diatom, this centric marine diatom, was again 25 and more % across the seasons, dominating our mud samples. Very easy to miss in certain cases. So we went back and made sure that we reviewed everything. So Lisa summarized 2016. This is Alyssa Thompson. Her research was 2016. So she summarized. So there's some of her Cimatosira belgica, and there's the mini discus in her documented material. And again, we published that in estuaries and coast, actually reporting significant decrease. In this graphic, so we have species richness, and this is at high and low tide at the same location that I took you a minute ago. And again, there was a significant decrease in species richness. Still the dominance was those 2 taxa that I mentioned, this Cimatosira belgica and Minidiscus. And from then on, I mean, I knew that there's quite a lot of variation at high tide. Of course, we did some interesting experiments. As I said, we will open it every 5 centimeters so we can see exactly what's in the water column after the tide is idle. But for some of the research that we continued, I really concentrated on the low tide when we had the mud, and we collected this mud, and the surface 2 centimeters of the mud that we were able to see and collect, and the significant decrease was there. And very quickly, when I was invited to talk, I finished the counts for 2019, 2020, and 2021. And I just looked at, again, for the 3 before, during 2016, the dredging, and after. I just looked very quickly at brackish, freshwater, and marine. In our taxa, we always identify them in terms of their preferences, and also benthic and planktonic. It was interesting, within year and between years, benthic and planktonic, from what we identified, there was no significant difference, but the increase in planktonic marine and the decrease of benthic, both you know, freshwater and brackish, was significant across the board. So just to summarize what we've done with some of the work, we knew that the dredging is coming, so we were trying to establish a baseline at this one location, and we were also measuring, of course, light, turbidity, salinity, and the response to all those different conditions. Then we looked at some, again, larger scale processes. We did some molecular work, we did whole community metagenomics and some other work that I'm not reporting today. But really, Diatoms were sending us a message. And this message, of course, it's quite clear that there's a change in the algal community. And this is what we published 2019 in the paper with Alyssa Thompson. And in preparation for this paper, I really got chills when I looked at, again, our site. And again, 2020, there is a signal coming from Cyanobacteria as phycoerythrin measure now that USGS is reporting. And again, this is all data prior to today. So I think what's happening probably with those benthic diatoms that were able to come to the surface, they're probably decreasing in numbers, and maybe cyanobacteria is coming to the surface and they're proliferating. There's some salt tolerant, but again, when I saw this, I was really shocked that, again, our latest data was showing almost no cyanobacteria in the surface material that we were collecting. But of course, we documented some forminium, some other motile cyanobacteria that probably are contributing to this signal that USGS is reporting. So it was a quick one, and I really want us to have some discussion on that I wanted to present to you. I hope I convinced you. In my lab, we looked at species and we protect species, and we relate that to many other governmental principles, and of course the Clean Water Act at this point. And I do stand behind that, identifying what's there and being consistent and really following taxonomy and documenting, of course, diversity indices, evenness and abundance is something that's still quite valuable. We've done, as I said, some metagenomics work. We will collect and we'll split into, if I have time, I might go into some of this work. We've documented more than 200 diatom species that we identified the species, and we have quite a lot more. I think when I retire, that's what I'm going to do, really go back and describe some of those taxa. We worked on motility. We did some very interesting experimental work. We'll collect mud and we'll bring it to the lab and we'll put different conditions. We never simulated them to move in the lab, but in the field, they will move upwards and they'll turn into this very nice brown layer on the surface. And then working on some physiology, we were able to actually prove with some of this work in Savannah River that with their physiology, diatoms actually really able to sustain some of the sedimentation that was coming with the tides upstream in the Savannah River. So my goals today were kind of, again, present what was done in the Savannah River three runs upwards, and it was really interesting to see the taxa in this protected environment are quite consistent, and still a lot of work to be done, especially with Gonfonema parvulum, as you saw. A little bit of applied ecology, so the microbial community was able to send us a signal that there will be a change in the community. We documented it within the diatoms with the increase of those chain forming, planktonic marine, and of course some of those small, reading the literature about minidiscus has been absolutely phenomenal. But there is a macro signal that potentially there's some replacement in the algal community with cyanobacteria. Of course, not probably just driven by what's happening within the community, but definitely maybe there's no more competition with benthic motile diatoms. And again, I highlighted few of the diatom species that we were able to record in our studies. So lots of literature and lots of funding. I know some of you are on call, and over the years, I'm really for all the stimulating discussions that we had, and thank you for all you've done. And again, there's some significant funding that helped for this 11 years of trying to go to all those locations and make sure that students are safe, because I didn't go all the time to there. So I'm very grateful for all the funding agencies. And It's tremendous amount of work that my students have done. So I really wanted to highlight what they've done in the last few years. And with this, I'm going to stop sharing and maybe open it for discussion.
Okay.
I'm not seeing
Thanks,
any-
Kalina. Julie?
Thank you. That was amazing. Very interesting to learn about your research. May I start with a practical question? So, Kalina, can you please, probably you said that, but maybe I didn't listen very careful. How exactly did you collect these mud samples? What volume of samples did you have? I mean, what kind of sampling equipment did you use? How you did it?
Sure. So when it's low tide and the muds are exposed, we will have certain amount that will just take with the syringe the top 2 millimeters, and we'll just collect that in triplicate. And we'll bring it with 3 hours from Savannah River. We'll bring it back to the lab, and depends on the day, we will do live counts immediately, or preserve the samples, because sometimes we'll bring again 20, 30 samples. We'll preserve the samples for live counts, and of course we'll digest and we'll do the live count, the clean diatom counts, and then the SCM work. Okay. So it was mud samples, surface mud samples. We had composite samples, but there was too many epiphytic. I don't know if you saw, there was some plants there. So for this is just the mud samples surface. We had epiphytic.
I was wondering how you split the epiphytes from the mud diatoms, this more epiphytic, because sometimes they could be mixed.
Epiphytic and so epipsamic is what we assume is there.
Yeah.
We didn't have much epiphytes. We didn't have much coconates or some of the others. We didn't have much in the mud. Only what was coming with the kind of the settling, how I imagine how as the water is retrieving, all these mud particles are serving as a seed for all this plankton, and then that's what left in some of the sediment. There was not much epiphytic. There was not much gomphonemoid there. There was not much coconates. There's not much achnanthes in the mud samples.
So you didn't struggle with those. They're very hard to identify.
Well, we struggled with some others, as you saw.
Yes, I see. This was absolutely amazing. Yeah, I have to research this genus. Okay, thank you.
I'm not seeing any other hands up. Oh, Mark. Would you like?
Get myself all put together here. There we go. Thanks, Kalina. Appreciate hearing about this extended project you've been working on for so many years, and extending Dr. Patrick's work as well. I have a question, kind of 2 questions about sort of that estuary and mudflat work. One is, can you give me a little bit of, you indicated this diatom mini discus, which sounds like something that's just designed to make diatomists frustrated. But can you give us just a little bit of information on the ecology of that? I mean, it looks to me like it would be a planktonic organism, but you're finding it in the mudflats. Does it live in kind of mucilaginous groups of little centric diatoms, or is it attached to sediments in any way, shape, or form?
Not as far as we know, and again, I'm struggling a little bit with what. So we went into trioculatus versus chilensis every time. So what we're finding, it was never more than 3 microns. Actually, I really will make students pay attention and really count on the good microscope, because they will meet it and they won't see it. It doesn't make any mucilaginous in the live samples. We didn't see it making any mucilaginous kind of collections. It's we saw it alive. We saw that there was something brown in there. We assumed that was the chloroplast. So we know it's living. It's I think it's just small, and just principles are selecting versus case selecting. It just proliferates quite a lot. It's in the plankton, and I think it's just caught and sieved into the mud samples. But I was shocked when I saw Stomatoxera belgica, and then I was seeing it everywhere. I was really those two were really giving me nightmare because I was like, that's not what I expected. Are we? I mean, and I was with the students. I know that that's where we collected, and that's what we saw, that were there. They're not making chains. I mean, Cyanobacteria belgica make chains. They're larger, probably they were caught there, no problem. I think there were just so many of them. Reading in the literature, it's planktonic, it's in the photic zone, and I think it's just proliferating very, very fast based on its size.
Okay, thanks.
We had
for that.
another question.
Yeah, one other question then. It was unclear, did you, with those estuary and surface mud samples, were you analyzing those alive, or were you preparing them and making diatom preserve mounts?
All of it. So I always told my students the trade-off of us sampling in one location 4 times a year, any possible collection we can have, is to really go deep into taxonomy, into analysis. Also, we found that 60 to 70% of what we saw was not alive. There was no chloroplast. So after we saw that 2011, for all our counts, as I said, when we bring the samples, the first thing we do is 300 units live diatoms. I have data for live and dead, for what we're able to identify. I showed you a little bit of the live material that we documented. Some we were not able to go to species, but we were documenting live to death ratio, because in the mud, there was quite a lot of deposition of those dead diatoms. And I was thinking, if I clean them and just report what was in the clean samples, I don't know if they were living, if they were actually altered in any way with of course, the sedimentation. So everything was counts live and clean.
How were you preparing the material for live counts that you looked at? Was it done in a I mean, was it a Palmer cell? Was it how did you do it?
Yeah, so first we'll look, so when we collect them in significant mud sample, so we'll just shake it and we'll take from the supernatant so we can just see what's in there. We'll put it into Palmer Maloney. The problem is in the Palmer Maloney, there was so much material. Another method that some students developed was, so we'll always look at Palmer Maloney and we'll look at just general density of live diatoms, and actually we'll document everything that was there. And then one student, so in the under 22 by 22 cover slip, you have exactly 0.02 microliters. Now we were collecting from an area and we were bringing it to volume, and we wanted to relate some type of numbers as density if we're collecting the same way over the years. So that flat slide with 0.02 milliliters from what we collected was easier to count. Was it meaningful for this Savannah River that I showed you? Of course it is, but again, it's very hard to put your mind around that. You're getting data from 0.02 milliliters of sample. That allowed us to have it in a single layer, and it was easier to see these mud samples.
Okay, thank you.
Jan?
So I'm going to pick up just off of what you just said. So what were the abundances of cells per millimeter squared or per centimeter squared from that quantitative work?
1.3 million, 1.4 million, and more.
Okay. So why do you think you're getting this shift from benthic to planktonic, and of course freshwater to more marine taxa? Kind of makes sense, but you're still not getting a plankton range. So why aren't you getting benthic marine taxa there?
I mean, I don't know why we're not getting benthic marine. There were some, so we had definitely lots of Pleuroxera in 2011 samples. We had, of course, Gyrosema, marine Gyrosemas. We had tons of Nitzias. I mean, you know, quite a lot of them that were typical marine rather than the freshwater. As I said, we did live collection, and then we did some experimental work so we can see who can move in the lab. Nobody moved. I think what's happening, it's just the volume. I actually spoke with a hydrologist about that because I just couldn't imagine what exactly is happening. So I showed you that the tide at this location, and by the way, we chose this location because historically it will have tides. Those tides prior to the dredging were 1.5 meters to 2. I think what's happening is just way more water, way more saline water comes, and it's mostly planktonic that's coming now on the surface. Now we're having 2 to 2.5 meter again increase in the volume above it. And then at the same time, the Savannah River, the mighty Savannah is coming. So all this fresh water are coming. And actually, I thought at this location we'll see a lot of fresh water. Even 2011, we didn't see that much. And that's actually published in the 2013 paper that we have there. So even if they were there, maybe there was so much going on. So those poor benthic moving diatoms, they were just not able to come to where we collected to the surface. And again, we're collecting, 5 by 5, 25 square centimeter area, again, to make inferences about what's happening. I think it's just the volume of marine water that was above the sediment that was happening.
Yeah, so it sounds like you do have the benthic marine taxa there. They're just rare compared to the planktonic forms, correct?
In our counts, again, we didn't.
Right.
I mean, the mud is not more than 5 to 10 centimeters that you have
Right.
there. So there's I mean, we can collect it all and we can look at it and in terms of numbers, as I said, everything that's over 5% is what
Right.
I'm reporting.
So can you, with having the densities and the relative abundances, can you calculate do you still have the same number of benthic the same number of benthic organisms in the habitat, and are you just adding on plankton because the water levels are more stable and you've got more of a lake-like versus an estuarine-like habitat?
Yes, as I said, I think it's been fun for me to go back to what the students counted, actually look at what they have. I mean, I can tell, there was some Nitziapelia and some of the freshwater were there, but most of what I saw was, and what we reported was marine.
Thanks a lot. Great talk.
Thank you.
Layla was next with her hand up.
Yeah, thanks, Kalina. It was as well about the difference between benthic and the planktonic and the increase of the planktonic one. As maybe it was, as you said, the increase in the force of the tide, but maybe as well the ability of the planktonic one to be in chain. Make them more resilient in this changing area. Is it possible?
Absolutely, because I showed you the Campylosera and the Sumatoxera chains. I mean, they were alive. We documented them. They were alive, and they were there. Obviously, they were pushed from the sediment and stayed there. But yeah, definitely big play from the chain diatoms. More Sumatoxera belgica than Campylosera, though. Very interesting.
And what about the competition with the Cyanobacteria?
I didn't think about this much. Of course, both Yelena and Alyssa Thompson reported Cyanobacteria mostly, as I said, for medium in the mud samples. I didn't think much. We had so many euglenoids. So we thought, oh, maybe it's increasing nutrients because we have euglenoids. We had, I mean, of course, many other, including chrysopods. But the Cyanobacteria filaments, as I said, my heart stopped when I saw USGS actually plotting phycoerythrin. I mean, they for all this time, and we've been monitoring that. Every time we go sample, we take a snapshot of what they have. Of course, you can go back and retrieve the data at any time. They never reported, and this is water column. This is a gouging. This is a gouge USGS area where they look at fluorescence and they detected phycoerythrin. It has to be coming from Cyanobacteria. We had some cryptophyta that might have phycoerythrin, but there were not as many as the Cyanobacteria. So again, I think what's happening in the mud samples, where you get light, those that are faster and because I think the diatoms were so many non-motile, those centric, those, I mean, I didn't report much. We had tons of rapid. They were not moving. They were just there. So those that were moving were maybe slow or lower in number as we assumed. And maybe something to the surface was coming, the cyanobacteria that were looking for light and photosynthesis.
Thank you. Nice talk. Thanks.
And please forgive me for massacring this name, but is it Dusika or?
Dushitsa. Hey, Dushitsa, good to see you. No problem. You gotta do, if you wanna ask a question, you gotta unmute. Dushitsa, gotta unmute.
Sorry. Hi, Kalina.
Hi, good to see you.
I'm happy to see you.
Congratulations.
Happy to see you too. I was wondering, did you collect only from the mud? And if yes, why did you choose to collect samples from the mud?
2011, we collected from everywhere. We collected at high tide water column. We collected anything attached, we did scrapings. And of course, at the mud. And the mud was something that was constant. Vegetation changed over the years. They had boat ramps completely renovating. So all our scrapings from there, they would put metal and there was very little diatoms attached to the metal versus the wood. So I have all this data that we collected, but what I found consistent was us going to this one location and in triplicate collected the surface of the mud. And I found that this is the only way I can compare over the years. I cited some of the papers. Each of the papers looked at all those different, of course, sampling collections, including, of course, epiphytic and planktonic surface versus 10 centimeters and so below. And I found that the only way I can compare for this 10 years was just look at what we have from the mud sample.
Okay, thank you.
Good to see you.
Good to see you too.
Are there any other questions? Kalina, did you, I know you mentioned that Ruth had diatometer data as well as some of your students. Did you see any significant differences in the community between the diatometer collections and the mud samples? Was it showing any kind of community bias or?
Yes and no. So again, the composite samples before Katie put the diatometers there, they were just, I mean, they were just showing ionoceous and gonphonema and some luticula. So Ruth Patrick reported luticula. At this site, gonphonema parvum was not as prevalent in 1956. It was there and it was reported. At this point, it was sensulato. It was, everything. But so if you remember, Ruth Patrick sampled the whole Savannah River, and the Academy actually shared some of the documents. Gonphonema parvum was a very important player in the Savannah River, and it's always been an indicator of high nutrients. And then when Katie put the diatometers, we had, I mean, as you saw, 16.3% gonphonema parvum. So it's a reference site. How are we reporting it as high nutrient taxon if it's there? So it opened all interesting things. And again, our goal was sensu stricto. What do we have in the United States after the Abarca paper and some of the other papers? We really wanted in this pristine location for 50 years to look into fine level taxonomy and really, report and hopefully send to Sarah North American gonphonema parvum, something that we have there. And Katie has done, as I said, tremendous work. I mean, to the point where even broken things, she will want to verify. So we knew, and obviously we counted them live. She identified them and isolated them, and now we're doing some molecular work. Hopefully that will help. But the answer is yes. So even in this 1956 diatometer and 2018 diatometer, gonphonema parvum became more prevalent. Is it just a better adapted taxon overall? Do we see it everywhere? I mean, the slides are there. Anybody can go back and check, and it's just amazing. But it was never as high in abundance in the 1956 counts that they had. And remember, they counted you know, 16,000, and I mean, it was an asymptote, right? So they counted in a very different way. That's why with Rob, knowing this, and of course, Jen, I think we counted what, 3,000, 5,000 after the 600. So in Kentucky, we'll count until there's nothing new. We never got to the, as you saw, 170 taxa that they had. I mean, Katie documented everything that was there. And we don't know if it was some of the unknowns that were also in the species richness, but definitely changed.
So much, yeah, it's a lot to think about.
That's
Any other questions for?
So fun.
Yeah, yeah.
Paula.
I had a question. Wonderful talk, Kalina, and as always, wonderful to see you. You've answered some of this already, but I was going to ask you, you brought up, the Gonfanima issue, like all the questions that have come up. Do you have a plan to take on some of these in the near future, or recommendations for which problem to tackle first?
So we can work on Onosha. You were supposed to come to Georgia so we can work on Onosha.
Pandemic.
I know. Definitely continuing the, so now we have this culture. So as Jen knows, what, 12, 13 years, well, it's been 15 years since I was at Michigan State. So we did this molecular metagenomics and whole community analysis and then counting. So I've been dabbling over the years into that. Almost everything that we have, we'll have half of it morphologically identified, and the other half will send for anybody that wants to work on molecular stuff. So to answer your question, Paula, I see myself definitely continuing to delve into some of those centric and planktonic marine from the Savannah River. I'm very into the Gonfanima parvulum complex with the interest from my current graduate students, and we're working on I don't know how many papers at this point. It's probably my fault because I don't have time to kind of finalize things. We still have some counts from 2021 that we haven't finished on the mud samples, and that's the next paper that I hope will have 10 years. I will recount again everything just with the newest taxonomy, because there was just a new mini discus paper that was published the other day. And of course, Cyanobacteria belgica, it's been reported as a tychoplankton, as a true plankton. It's in the mud. So obviously there's quite a lot of other patients that we can report there. Plus the sizes that we're finding in the Savannah River. They were, again, way larger than some of the reported literature. Cyanobacteria belgica of 24 length was something very, very unique. I was like, that's maybe not, but it is, it looks like. So yeah, so lots of interesting things. Jen, you have Julie, I think Jen has a question.
Yeah, I was waiting for Julie, making sure you were done.
Oh, sorry, I thought you'd take control there.
But I was just going to follow up on Paula's, and then the idea of the parvulum exilisimum issue. And you said you did some molecular work earlier, and does it link in at all to any of the work that others have done, like Regina? And you said that it didn't match, if I remember correctly, anything, but it was similar to other things on that gradient.
Oh, we're going to report a brand new 23S genome information that is not in the database. The chloroplast genome, the RCDL genome that we have, is 100% matched with what's considered their epitype of Gonfanima parvulum that they have there. So we actually have data for 2 genes. So one was perfect match, the other one there was nothing to match it to in the database. So that's what we're working on right now. And then all the morphological work that Katie did will be complementary to that. And that's all from this one location, protected for 50 years. I regret we didn't have funds to measure nutrients from on Katie's work, but I assume SRS hopefully has some nutrient data there. just because maybe there's some change in nutrients. So that's one thing as I was preparing, we just didn't do nutrients. We did just YSI measurements at the site, but we didn't do nutrients. And I kind of regret, did I know that she's going to isolate it and we can do all these amazing things with the culture? But looking back, I wish we did nutrients.
Yeah. So on a more serious note, I did not like your comment about you saying when you retire.
Oh, I mean,
you said that as an aside. When I retire, I'm going to work on it. You're not allowed to think about that yet.
oh, thank you, Jen. But yeah, I'm old.
If by old you mean fabulous, then we buy it.
Thank you. Thank you. Thank you.
Jan, my internet connection is a little bit problematic right now, so I may be missing questions if you see anything.
I don't think there are any more questions, and we are
No.
getting to the top of the hour, so we can adjourn for the rest of the day. And I'm sure if anybody has any follow-up questions, we know how to track Kalina down and ask her those and ask her how her grown children are doing. But anyways, thanks, Kalina, for coming and speaking with us.
Yeah,
Thank
so
you.
thank you, David, so I can keep my young children picture there so people think that I'm young, but
Yeah, keeps you young.
yeah, thank you so much, everybody. It was fun.